You know, every single year, infections that we used to consider minor are becoming life-threatening. And the reason why? Well, it's because bacteria are getting smarter. They're evolving and building up resistance to even our most powerful last-resort antibiotics. It's like this silent pandemic unfolding right under our noses, and it threatens the very foundation of modern medicine. But what if we could finally crack their code? In this explainer, we're diving into some incredible research that uses artificial intelligence to do exactly that, to finally get a step ahead in the fight against these superbugs. So our whole investigation here is built on a massive, and I mean massive, global effort. Scientists gathered thousands of bacterial samples from all over the world and sequenced their entire DNA. The goal was basically to create a genetic blueprint, a map that could pinpoint exactly what makes these superbugs so incredibly tough to beat, and to see how they're evolving on a global scale.
And right at the heart of this whole crisis is resistance to drugs like meropenem. You need to think of meropenem as the last line of defense. It's part of a class of antibiotics called carbapenems, the ones doctors turn to when they're dealing with the absolute worst, multi-drug-resistant infections. So what happens when this drug stops working? Well, doctors are left with almost no options. And that's exactly why figuring out how bacteria are beating it is so unbelievably critical.
Okay, so how exactly did they crack the code? We're going to follow the evidence, just like a detective story. First, we'll introduce the main culprits in the superbug crisis. Then, we'll map out the mountain of genetic evidence they left behind. After that, we'll bring in our AI detective to sort through the patterns, reveal the key genetic clues, the smoking guns, and finally, uncover the vast web of resistance these bacteria have been building. All right, let's meet our two prime suspects. Our investigation focuses on two of the most dangerous superbugs out there. You've got Klebsiella pneumoniae, we'll call it KP, and Acinetobacter baumannii, or AB for short. They're both part of this notorious group called escape pathogens, which are a huge cause of hospital-acquired infections. And what makes them extra tough is that they're gram-negative bacteria, which means they have this extra outer membrane that basically acts like a suit of armor, making them naturally hard to kill. And like we said, these bacteria are fighting back against our very best weapons, the carbapenems. These drugs are basically the special forces of the antibiotic world. They're designed to take out a huge range of bacteria by stopping them from building their cell walls, which makes them just fall apart. But our two suspects, KP and AB, have figured out ways to dismantle this weapon. They've evolved defenses to neutralize this super powerful class of drugs. Okay, so now let's dig into the evidence. Imagine having access to this absolute treasure trove of genetic data. It's amazing, but it's also like trying to climb a mountain. How in the world do you find the few crucial clues that matter when they're buried in the DNA of thousands of different bacteria? And the scale of this analysis is just wow. They looked at the complete genomes of 2,411 KP samples and 375 AB samples. We're talking 2,786 full genetic codes. This global dataset gave them a view that no one's ever really had before, a chance to see how these pathogens are building their defenses all across the planet. And when you lay out the data like this, you see something fascinating. Our two culprits have completely different playbooks. Look at KP. Its main strategy is target alteration. A massive 64% of its key mutations are focused on changing the very part of the cell the antibiotic is trying to hit. But then you look at AB, and it's all about antibiotic efflux. It builds these little pumps to literally throw the drug out. A whopping 60% of its genes and a staggering 67% of its mutations are dedicated to that one strategy. So they're both resistant, but they're getting there in totally different ways.
So here's the bottom line, the crucial difference. KP's strategy is basically to change the locks. It modifies its own machinery so the antibiotics key doesn't fit anymore. AB, on the other hand, it's more like a bouncer at a club. It doesn't bother changing the locks. It just builds these powerful pumps to physically throw the antibiotic out before it can even get to the dance floor.
Now, with thousands upon thousands of genes and mutations to sort through, finding the ones that actually matter is a classic needle in a haystack problem. And that is where our AI detective comes onto the scene. The researchers used machine learning to essentially train a computer to become an expert genetic profiler.
And here's how they did it. First, they just dumped all the genetic data into the system, whether thousands of different genes and mutations were present or absent in each sample. Then they used some clever statistics to filter that down, to zero in on the features most likely to be important. Next came the training montage. They trained several different types of AI, teaching each one how to tell the difference between a resistant and a non-resistant bacteria. And finally, they held a competition to see which AI model was the best detective of all. And well, the winner was pretty clear. As you can see from this chart, the different AI models all had some success, but one of them, called a support vector machine, or SVM, just consistently blew the others out of the water. It's really good at sorting complex data into clean categories, and it nailed this task, achieving an incredible test accuracy of 96% for KP and 93% for AB. This wasn't just a lucky guess, this was a seriously reliable tool.
So, with the SVM model crowned as their top detective, the researchers were ready to move on. They now had this powerful algorithm that could cut through all that genetic noise and point a big flashing arrow directly at the features that matter most in the fight against meropenem.
And this is it, the moment of the big reveal. Using that super accurate AI model, the researchers were finally able to identify the specific genes and mutations, the smoking guns, that were the absolute strongest predictors of resistance.
For Klebsiella pneumoniae, the AI pointed to these 4 key genes. Now, the first two, block KPC2 and block KPC3, those are the real heavy hitters. They produce these powerful enzymes called carbapenemases that literally just chew up and spit out our best antibiotics. But what's also super interesting is that ACC gene. It actually provides resistance to a completely different class of antibiotics, but its presence is a huge red flag for meropenem resistance too.
Now for Acinetobacter baumannii, the story is a bit of a mix. Its primary weapon is the BLA-OXA23 gene, which is another type of antibiotic-destroying enzyme. But the AI also flagged some critical mutations. That mutation in OPER-D, that's like bricking up a doorway into the cell so the antibiotic can't even get inside. And that GYRA-K mutation, again, it gives resistance to a whole other class of drugs, hinting that there's a much bigger conspiracy going on here.
And that leads us to maybe the most important insight of this entire study. Antibiotic resistance isn't just about one gene doing one job. It's a complex network, a web of interconnected defenses all working together to make these bacteria practically invincible.
And its visual just illustrates that web perfectly. The researchers used a technique called association mining, which is basically a way to find co-conspirators, to map out how all these clues connect. For KP on the left, you can see how genes like KPC2 and that AAC gene often show up together, leading to meropenem resistance. And for AB on the right, it's that killer combo of the OXA23 gene with mutations in OPRD and GIRA that creates this impenetrable fortress.
But the problem gets even bigger. This incredible map shows that resistance to one drug doesn't happen in a vacuum. If a bacterium is resistant to carbapenems, that's the big pink cluster in the middle, it is highly likely to also be resistant to tons of other antibiotics. See how it's connected to those yellow, green, and purple clusters? Those represent completely different drugs. It's a phenomenon we call co-resistance.
And here's why this is so terrifying. The genes that give bacteria these powers are often bundled together on these little mobile pieces of DNA that they can actually pass around to each other. So when a bacterium gets the weapon to defeat carbapenems, it often gets a whole toolkit of other resistance genes for free. We saw this with the ASAC gene in both bacteria and that GRA mutation in all the resistant AB strands. This is what creates these multi-talented superbugs that are resistant to almost everything in our arsenal.
So, let's bring all these clues together and wrap up the case. We've learned that our two culprits, KP and AB, have different main strategies for defense. We've identified the specific genes, the ringleaders, that are driving this resistance. We've seen just how powerful AI can be in picking these needles out of the haystack, and most critically, we now know that this isn't a single gene problem. It's a network problem, a web of co-resistance.
What this research really gives us is a powerful new map of the enemy's battle plan. By understanding not just the individual genes, but the networks they form, we can develop faster diagnostics, smarter ways to track outbreaks, and maybe, just maybe, new drugs that can outsmart these defenses. So the question isn't, can we understand them anymore? The question is, how quickly can we act on what we now know?